We're set to resume with an internal speaker. It's a pleasure to introduce Daniela Schlatzer. She's a senior research staff member here at the Case Center for Proteomics. Dani comes to our center with abundant industrial experience based out of GSK at RTP, North Carolina. She is specialized in biomarker discovery with specific emphasis on label-free approaches for discovery proteomics. She's also accomplished a lot in the quantitative aspects of quantitative and validation aspects of discovery work. Her interests also include analytical method development for different issues that the center tackles. She is at the forefront of our center's efforts towards label-free discovery proteomics. Once again, it's a pleasure to introduce Dani Schlatzer. Take it away, Dani.
So thank you very much for inviting me to speak today. And I'd like to thank the external speakers for some wonderful presentations this morning, and I look forward to those in the afternoon. They've been very interesting. We're going to switch gears a little bit in that I'm going to highlight network analysis for putative biomarker discovery and method validation in the early stages of method development. I'm going to be using existing networks and not sort of trying to find new novel networks available. So this data is going to be using many of the commercially available network analysis data visualization tools such as Metacore, Pathway Studio, and IPA. So I'd like to, with regards to the outline, I'd like to highlight the platform that we use for quantitative proteomics in biomarker discovery here, which is label-free protein expression. It's a shotgun proteomics method. You've heard many people this morning talk about that. And I'm going to provide two examples. The first being, marker's gone here on this one. The first being a plasma biomarker discovery for idiopathic pneumonia syndrome. And the second, showing how I'm using these network tools in the early stages of method development for a label-free protein expression of primary dendritic cells from human subjects. So with regards to the label-free analysis workflow, whether it be cells or plasma, each sample is individually prepared. For plasma, we do a top 7 or top 14 depletion of the most abundant proteins. For cells, we need to generate we need to lyse the samples and then put the samples in a buffer that's amendable for solution-based digestion. Here's a simple comparison here just for the purposes of explaining the workflow in which you have a control and a test. Each sample is individually digested with trypsin and/or lyse. And then that solution is directly analyzed via LC-MS using a high-resolution instrument such as the Orbitrap or the LTQFT. Once we've acquired this raw data, we need to translate it and transform it into something that we can do statistical analysis on. So this involves some significant data processing that we use using the Infochromic software that we have in-house, in which we generate peak abundance or generate chromatographic peaks for individual peptides, and we cluster that across the data for control and test samples, such that a peptide is now seen as a row with its individual peptide intensity per sample. And this becomes a fairly high-dimensional data set at this point, where we can actually track thousands of peptides across many samples. And so because of that, we need to apply higher-level statistical analysis tools so that we can ensure that we're assigning the most appropriate significance to the data. So that we can generate a list of peptides and those proteins from which they're derived from to take further into network analysis so that we can understand not only the this change in the fold change or the change in the intensity of these proteins, but how they relate in terms of the biology to the disease, so that we can identify biomarkers that not only are statistically changing based on a value of abundance, but also their biological interest. So for the first example, I'm going to highlight a recent clinical study that we've completed, and it's looking at idiopathic pneumonia syndrome. And idiopathic pneumonia syndrome is a complication of allogeneic stem cell transplantation. And probably all of you know stem cell transplantation is an important therapeutic option for both malignant and non-malignant disorders. However, there are a number of limitations to its use, those being regimen-related toxicity from the conditioning, which can lead to infection, hemorrhage, multi-organ failure, graft versus host disease, as well as pulmonary toxicity, which can either be acute or chronic. And idiopathic pneumonia syndrome falls into this category as an acute lung injury without the presence of bacterial infection. Its incidence is about 10 to 15%, and it affects the pediatric population. And while it's it's a rare complication in these patients, it is extremely fatal, with approximately 70 to 80% mortality rate in these patients. And the onset is typically 10 to 21 days after stem cell transplantation. So our collaborator, who is a pediatric oncologist here at Case, Kenneth Cook, has spent a lot of time developing mouse models of this disease to study it in the lab. And what he has observed is that there is very similar histology and functional changes that occur in these animals in the lab that recapitulates many of the changes that are seen in humans, where you have dense mononuclear cell infiltrates into the lung and bronchial tissue, as well as very high levels of TNF-alpha and huge amounts of inflammation and pneumonitis in these animals. And indeed, when treating these animals with an anti-TNF drug, you can reduce the severity of the disease. So he's been able to translate that work into a clinical trial for human subjects using Etanercept, which is also known as Enbrel. It's an anti-TNF-alpha drug. And what you can see here is this is bronchial lavage protein levels. And you can see that at diagnosis, you have very high levels of the total protein levels in the bronchial lavage fluid. Bronchial lavage is just basically is a fluid that's extracted out of the lungs after a flush of saline. And with treatment, you get a decrease in the overall protein levels, as well as many of these key inflammatory cytokines. You can see decreases after treatment. And some of these patients do have recovered from this complication with using this treatment. So it's quite encouraging. So using much of his clinical trial work and also the insights that he's gained from the laboratory, he's started to define IPS as no longer as an idiopathic clinical syndrome, but one with a potential disease pathway in which you have both the adaptive and innate immune systems becoming involved. In terms of the gut, in terms of the inflammatory axis, you have the gut being compromised due to bone marrow conditioning, leaching out endotoxins like E. coli or like LPS, not being properly filtered through the liver. And this LPS goes on to sensitize macrophages to start secreting TNF-alpha, and a dysregulation of the immune system occurs, and it ends up actually attacking the lung, as well as donor T cells from the bone marrow transplant secreting interferon gamma and also priming these macrophages. So while he's provided a framework in terms of the disease, there's a lot known about any other inflammatory mediators that could be involved. So we embarked on a label-free protein expression study to expand his disease pathway. And in that, we looked at 22 samples that were analyzed from stem cell transplant patients in two groups, either a non-progressor, these patients got a stem cell transplant, but didn't develop IPS, as well as patients who developed IPS after the stem cell transplant. And plasma was drawn at blood was drawn at day 0, as well as day 14 for those that non-progress. That's the median onset of IPS, or at diagnosis for those, at the day of diagnosis for those that got the sample or got IPS. So all of those samples were individually put through the pipeline, and I wanted to talk just a little bit more in detail in terms of the data analysis after the raw data has been acquired. As you've heard earlier, it's very important in this type of data to properly analyze and normalize the data due to the high degree of variability that can be seen. So the first thing to do is we need to basically process the data using our Infochroma software, in which you generate intensities versus samples. We do that by generating a peak list with masses of individual peptides, and then we generate chromatographic peaks for those peptides. And then for those masses that we have a sequence assignment, we will put that in the spreadsheet as well. Once we have the spreadsheet, we need to perform data QC before statistical analysis to ensure that we're progressing the most robust peptides forward for this type of work. So we need to filter core quality peptides, those which received a low mascot protein score, as well as replace missing values that can often be found in this data. And then finally, we need to normalize the data so that we ensure good normality and stabilization of variance across the data set so that we can move forward for statistical analysis. And once we've been able to do that, we then go ahead and perform ANOVA, PCA, and adjust for fault discovery rates. And much of this work has been spearheaded by John Dizard and Rob Ewing in our labs. So with regards to the summary statistics for this study, we used a two-way ANOVA that looks, when we chose a two-way ANOVA because we wanted to look at changes of proteins across time and status, and this is defined as an interaction effect. And in the study, if you recall, we have status and time, both control at 2 different time points and IPS at different time points. So for the data that we took forward, we had over 1,253 peptides that received a peptide assignment, and after we did our QC filter, approximately 1,100 of those passed the QC filter and were analyzed by the two-way ANOVA. And when looking at the interaction effect, you can see that we have very significant changes for interaction, with almost half of our data changing, with an adjusted p-value of 0.1. These represent peptides that are up or down regulated, and this is to be expected because these patients are extremely, extremely sick at the time of diagnosis, so they should have big changes in their proteome. And this corresponded to over 100 proteins that changed with interaction effect. When we take those 100 proteins and put them in engineering pathway, we can see that they're hitting the top 6 functions are hitting a lot of things that make sense in terms of his proposed biology, and that antigen presentation, cell-mediated immune response, humoral immune response. These things all make sense in terms of problems, dysregulation with the immune response occurring. So this was extremely encouraging. And when we go to look and put some of these proteins on pathways, we also see that we can see a footprint of TNF-alpha and IL-6 signaling. If you recall, he believed that, and he had shown in his ELISAs, that there were increased levels in these inflammatory cytokines. And indeed, while we didn't identify these, we did identify many good acute phase response plasma readout proteins that correlate nicely with high levels, that their abundance correlates nicely with high levels of inflammatory cytokines. So you can see that, plasma acute phase proteins that should be going down with increased levels of TNF-alpha are indeed going down, and those that should be going up. We have a fairly nice correlation across that group as well. As well as we feel like we're potentially expanding his disease pathway. And here's a couple selected proteins. All 3 of these are involved in pattern recognition, and 2 of them have been fairly well documented in terms of antigen presentation. MBL2, you can see, has slightly higher levels. This is looking at a protein level view of the data. It has slightly higher levels at day 0. These patients, if you look at them, they look the same. Clinically, then those with control. This is the median value across that protein or peptide. As well as Lumican appears to have slightly higher levels at day 0 than day 14. And then lipopolybinding protein has slightly higher levels at day 0 as well, but then very significant increase at day 14 for this protein. And when you start to think about how these proteins could be interacting, you see that there has been a lot of evidence suggesting that many of these proteins are involved in toll-like receptor signaling. LBP is a very well-documented antigen-presenting molecule that initiates sort of the cascade of events that signals transcription of pro-inflammatory cytokines like TNF-alpha. With Lumican and MBL2, Lumican just recently being cited to interact directly with TLR2 and TLR6. It co-IPs with CD14, and Lumican knockout mice are hyporesponsive to LPS-induced shock. And MBL2, while it's classically known as being involved in activating complement, it also has been shown in interaction studies to interact with this molecule. So this is quite encouraging data and previously not known with his disease. So we wanted to look at these selected proteins at an individual basis and see what's going on with individual patients. And when you look at time 0, day 0, the red here is patients, IPS patients, and the yellow is control. You can see that there are 3 patients that have significantly higher levels, approximately a 40% increase for Lumican. And when we unblinded the study, we found that these 3 patients were the patients that actually responded to anti-TNF therapy and had good outcome. MBL2 has a similar pattern, although not as significant, in that you can see that overall these 3 patients have this higher level. So it does make one wonder, could these patients already be sensitized to an inflammatory response that can cause a dysregulation? So very interesting data. And then when we look at LBP, this is just verification. This is just a verification study that has just recently been validated and is in IMPRESS. For LBP, you can see that in the label-free work, we are seeing approximately a 5-fold increase when comparing control versus IPS at day 14, and they've expanded the sample set to a larger cohort, looking at and looking across those patients, and you can see control at day 14 versus IPS. They're seeing approximately a 3-fold increase. So this is very nice cross-validation of individual work. This is an ELISA here. So with regards to this component of my work, we would like to validate Lumican now next via a Western. And so we'd like to do a verification via a Western or ELISA in a larger cohort of patients to see if we can see this trend, as well as investigate a knockout mice that is a knockout model of this disease, which is available here at the university. And then finally, we'd like to perform a verification label-free analysis in a larger cohort, so further depleting the sample of depleting additional 7 proteins to see if we could recapitulate some of the observations that we made in the smaller study. So next, I'd like to move on to just some method development that I've been doing, focusing on the very early stages of a project. And this is looking at dendritic cell proteomic analysis for quantitative proteomic work. And the goal is to investigate proteomic changes in primary dendritic cells across different HIV populations. And the sample is basically dendritic cell selection purification by this Miltenyi-Beat approach. For those that are familiar, it's very much like an IP. And they're pooled from 100 mils of whole blood. And this purification typically yields about sub-e to the 6 cells. So it's for a shotgun proteomic experiment that's working at fairly low levels. So, and after the lysis and detergent cleanup that we need, we are left with about sub-microgram levels of protein. So this is fairly low amounts of protein to do this work with. So the challenge is can we do quantitative or proteomic analysis of low protein concentrations? And if so, can we quantify proteins that are of biologic interest? So this is sort of the first pass of some of this work, and you can see that we are indeed getting a nice distribution and good overall peak intensity. This is LCMS data of MDCs at 633K and PDCs at 132. That's the starting. So it looks like we're getting very good digestion out of this sample at this point, these samples at this point. And when you look at the shotgun approach, these are non-fractionated samples, so they're just basically put on. There's no fractionation up front. We're obtaining over 400 non-redundant proteins from the MDC and over 200 non-redundant proteins from the PDC sample, with many MHC proteins, immune-related response proteins, that are of interest, as well as an interesting myeloid differentiating antigen protein that is of biological interest as well. And when we look at, there were some questions in the beginning of how well we were lysing the cells with our protocol. So we used the cellular location function in the pathways, the software tools that we have in IPA, to just see the distribution of proteins that we identified and where they're located in their cellular compartment. And we can see that we're getting a pretty good representation in the plasma membrane, to be expected of what you typically get when you do just a straight up lysis, as well as nice distributions across other regions. So it was encouraging that we felt like we were lysing effectively. And when we take the proteins that we identified and put them again into IPA, we can see that we are getting a number of proteins that are immune-related functions. So we're seeing a lot of things that biologically our investigators are going to want to track when we do the quantitative assay. And that's very encouraging, and the distribution seems to be fairly similar across both the cell types, both cell types of dendritic cells. And so with regards to doing some additional work, we want to determine the reproducibility of the lysis protocol that we've developed for dendritic cells. And then we'd like to perform a pilot of healthy control DC subsets comparing the presence or absence of TNR ligand in an overnight cell culture. So this would just be treating the cells overnight with this ligand and seeing if we can see differences in those immune proteins that we want to track. And then finally, if this pilot works well, we want to take this to a clinical study utilizing label-free of the DC subsets in both HIV versus HCV and HCV and HIV infection, to see if the HCV infection affects MDC and PDC numbers and function. So monitoring the biological changes with the two different types of populations. So finally, I'd like to acknowledge everyone at the center, and especially Mark Chance, our director, our collaborators that are always really important in doing this type of work, because they help you understand the biology and why you're doing what you're doing. And as well as John Udez and Rob Ewing and Gaurav Rana in the bioinformatics section for a lot of their important insight and data analysis for this work, and also Interactome for their software package, as well as our funding sources. And that's it.
Thanks, Eddie, for a nice presentation. We have room to take a question at least. Please.
Regarding your second project, HLA genes are highly polymorphic. So I was wondering how this is going to affect your detection of differentially expressed markers.
So yeah, that's something we'll have to investigate as we go and look at multiple different patients and see how consistent the peptides we identify are. That we don't know. This is just very early work on just a couple samples.
Okay, if not for further questions, let's thank Denny again. Thank you, Denny, for a nice presentation.